And welcome to this month's edition of the Clinical AI Interest Group of the Alan Turing Institute, specifically the Genomics AI Specialist Interest Group. We're very pleased to be joined by Jun Cheng, who's a research scientist at Google DeepMind. He led the AlphaGenome project at Google DeepMind and was the first author on that groundbreaking paper in Science. And we are very lucky that he's here to talk about AlphaGenome. His broader interests include training genomics foundation models capable of addressing diverse and critical challenges in the field. So I'm sure you have many questions to ask about him on that front as well. Jun, I'll hand over to you. Thank you.
Thanks, Alex. Thanks everyone for dialing in. It's nice to present here to the Genomics AI audience of NHS. I hope you have your lunch with you, because feel free to have your lunch while listening. Yeah, today's topic is about AlphaGenome. It's a model to advance regulatory variant effect prediction with a unified DNA sequence model. So previously we published AlphaGenome, which is focused on the coding variants, and AlphaGenome will be focused on non-coding variants within the gene body, but also like regulatory variants in the intergenic region. So we all know that human genome is a very complex book. It's really difficult to read. So we think that a natural way to read the human genome is actually through machine learning or AI. Yeah, we talked about that previously. We have AlphaGenome on the coding region, but the non-coding part, previous models like Informer or BPNet, they have, they predict gene expression from sequence, but there's certain limitations of that we'll talk about later. Gene expression is really multi-step process. It's really complicated. From DNA, you have DNA modifications, chromatin accessibility, and many things, transcription factor, many things that regulate gene expression. And after the transcription on the RNA level, you have RNA splicing, polyadenation, many like post-transcriptional modification steps. And on the translation part, you get to proteins, and many of the variants even affect on protein function. So previous models like BPNet or Splice AI, they are relatively short sequence resolution, high resolution, but very short sequence window. What that means is that in order to predict something, you input the DNA sequence as input, right? And for these previous models, the sequence they take into consideration is relatively short. And even though they predict profile at very high resolution, for instance, transcription factor binding, they can predict the binding profile at base pair resolution, which is great. And on the other side, you have Informer or Bose model. They take relatively wide input sequence length, up to 500,000 base pair for Bose. But then the resolution is very low. For instance, for Informer is 128 base pair, and Bose is 32 base pair window. They're not a 2 base pair resolution. So with AlphaGenome, we solve these two problems with one model. The model takes 1 million base pair sequence as input. This roughly corresponding to the majority of the regulatory elements. So if you think about the enhancer to the gene, 1 million base pair will cover most of the enhancers. And we also predict gene expression profile or other profile like transcription factor binding at base pair resolution. And the technical detail, I'm going to skip that technical details, but underlying is a UNet model. It has an encoder and decoder and transformer layers in between. And in the end, it predicts many different outputs, including RNA-seq, cage, and DNase ataxic, transcription factor binding, and many splicing outputs. It also predicts contact map, even though at lower resolution. So overall, you can think of the model as taking 1 million base pair DNA sequence as input, and that's the only thing the model takes as input, and it predicts all the different things that you can measure experimentally about that sequence. For instance, when you do RNA-seq, you have an RNA-seq track profile, coverage along the sequence. That's something we predict. So with ATAC-seq and other things, with splicing, we predict where the splice sites are and the usage of the splice sites. For instance, the usage will be 0% to 100%. And we also predict the splice junction. This is kind of the sashimi plot. You can plot with IGV. You can see which exon is being skipped and which are the alternative splicing junctions. And yeah, overall, the model has 450 million base pair parameters. This is a small model in today's terms compared to large language models. But we can train the model in a very short time, like within 4 hours on our TPU hardware. And it's actually 50% cheaper to train than the previous model, Informer. Just to give a high level what the model is predicting. So this is 1 million base pair window. Do you actually see my pointer? Maybe not. Is there a way to show my pointer? Okay. So on the right is the 1 million base pair window. You can see like many genes within this window, and the model predicts the RNA-seq profile for the positive strand and negative strand, let me just check whether I can show my pointer. Okay, I give up. So the ATAC-seq profile as well, and bottom here you can see contact map predicted versus mirrored contact map. And on the left side is a zoomed window of one specific gene. This is LDLR gene, and the here we show the predicted splice size as tracks and splice site usage, and also importantly we can predict the splice junctions, exactly what are the junctions being expressed, and the RNA-seq profile, RNA-seq coverage as well. You can see very clearly the different exons being predicted on the RNA-seq coverage and the different introns being predicted on the splice junction. Here's some example of the contact map. On the first row are the experimental mirrored contact map, and second row is some previous method, and the third row is AlphaGenome. Got it. The third row is AlphaGenome, and you see the correlation number are generally higher with AlphaGenome. So here's the track prediction performance overall overview of all the different tracks. For instance, splicing RNA-seq, DNA accessibility, histone modification, contact map, and TF binding. And you see in general the raw profile prediction is generally better with AlphaGenome. What that means is that you can think each of these prediction as to predict what experiments are mirroring, right? For instance, RNA-seq coverage, you're trying to predict how well you are recapitulating what RNA-seq experiment is mirroring the coverage. And these metrics generally tells you how good you are on predicting this coverage profile, right? Where the exon coverage, where the intron coverage. For instance, if some exons being skipped through splicing, the expression level for that specific exon will be lower. And that's something we're mirroring. And we show that this is generally better than previous model, even though we have one model and we're picking up the best model for each of these category. And this is one example on the gene expression. This will be gene expression level summarized by per gene. And you see that the Pearson correlation is generally higher for AlphaGenome, and each dot here is one specific condition, one specific cell type. So overall, we predict around 300 different, 300, 400 different cell types or tissues, including GTEx tissues and some encoded cell types. And yeah, so another technical detail is about distillation. I probably skip that. Regulatory variant, this is probably something that a lot of you are interested in, how well the model is doing on predicting variant effect. We break this down to different categories. We start with RNA splicing, predicting expression change, and DNA accessibility and TF binding. These are mostly regulatory. Expression can be regulatory, and also, for instance, variant can come from promoter region or enhancer region, and splicing will be the variants on the gene body that affect RNA splicing. We look at the clean bar. This is one of the widely used variant pathogenicity label, and we predict if a variant is causal causally linked to splicing, and we are generally doing quite well. GTEx splicing outlier, this a rare variant that cause aberrant splicing change, like cryptic exome or cryptic splice size. And we also have the expression. We focus on mostly EQTL benchmark, and each EQTL, we break this down to a few different tasks. The first is predict the direction of EQTL, whether the EQTL is regulating the gene higher or lower, or the causality of the EQTL, if this is a causal EQTL or not, and enhancer to gene linking and polyadenation variant, that variant affecting polyadenation of the transcript. And DNA accessibility, we have some benchmark telling you, if the variant is associated with more accessible or less accessible of the DNA, and transcription factor binding as well, similarly. Yeah, so on the RNA expression, performance, the model we benchmark with Splice AI, which is quite widely used, and Pangolin as well. So one thing to note is that the AlphaGenome predict both RNA-seq coverage, splice site usage, and splice junction. I think this is the only model that does all of this. If you think, like for instance, with this as one example, this is one example from GTEx. One mutation close to the exon caused exon skipping, and this is the predicted splice junction for the reference. And the alternative, you see that we predict the exon being skipped with the mutation. And with the observed RNA-seq coverage, you also see that with the mutation, the coverage is roughly half compared to the wild type. And splice site basically telling you that the splice site is deactivated for that exon being skipped. Here's another example. The mutation caused actually disrupt the canonical splice site, but then created alternative cryptic splice site in the exon region, in the intronic region. And we see that on the RNA-seq coverage, the exon being elongated, right? You have a bit of hair elongated, and the new splice site being predicted. And the junction also tells you that here's a new junction, which we don't see in the wild type, and the wild type junction is no longer there. And we do in silico mutagenesis. This is a model interpretation method just to tell, that by doing this kind of prediction, what are important for the model to make the predictions, right? For splicing, we know that there are splicing donor and acceptor. These are quite important for splicing, but we also know that there's a polyadenation track and branch point, and all these features are being captured very well by the model, meaning that these letters, height of the letters, basically telling that how much the model is taking use of this information. And you see that the model is actually pay a lot of attention to the splice size and also some other important features we know. Yeah, on the splicing performance, we overall, as we mentioned very briefly previously, we benchmarked on splicing outlier, as splicing QTL and ClinVar benchmark. That overall the model is doing quite well on all these different benchmarks in terms of predicting which variant is causal related to RNA splicing. So here are two like deep intronic cryptic splicing examples. You find some more examples in the paper. I think this is one of the type of variant that people are paying more and more attention to them. So these are deep intronic variants, but what they do is the mutation of these variants, they create a cryptic exome in the middle of the intron, right? So a lot of times this causes problem for the transcript because this may cause frame shift and leads to decay of the transcript. And you can see that the model predicts very well the cryptic junction, cryptic splice size, and the cryptic exome coverage. Do people have questions? And we can take questions in between. And if not, I can continue. So previously we talked about RNA splicing, and now we go to a bit more regulatory variants on DNA accessibility. This variant affecting how much the DNA is accessible, because transcription happens when the DNA is accessible, right? If DNA is not accessible, typically transcription will be repressed. And here we talk about variants that are affecting the accessibility and transcription factor binding. And we look at a few benchmarks here. The first one is chromatin accessibility QTL. And this we assess at a few different ancestry groups. And on y-axis is average precision on predicting if a variant is causal for causing like DNA accessibility change. And you see that AlphaGenome is generally doing quite well on this task. And here's one example of CRQTL in African group. And you see that this is observed effect size and predicted effect size. And in general, they correlate pretty well. On the coefficient task and predicting the effect size change, on this effect size, AlphaGenome is also doing quite well. So here's one concrete example that one of the variants that affecting RNA-seq coverage. So if you do RNA-seq, you typically see, so this is the lost exon. Actually, this is negative strand gene. This is lost exon. You see with the blue is the wild-type RNA-seq coverage. You have quite some expression for that gene. And with the mutation, which is coverage shown in the red line, and you see that with that mutation, the gene is basically no longer expressed. And if you look carefully, this gene is a gene that affecting expression, but it's also a splicing variant, actually, because it caused some problem in the splicing. Therefore, the gene is the transcript is destabilized, and therefore on the coverage side, you see that the gene has much less coverage. Yeah, so here's some other examples of eQTL coefficient. This is to predict, previously we talked about if a variant is causing expression change, and here we talk about by how much, right? The coefficient, the strength of the eQTL. And in this case, we predict the coefficient quite well, even though that I have to say, the effect size, we systematically underestimate a bit the effect size compared with the observed ones. And if you stratify the sign prediction by distance, this is to predict like if the EQR is upregulating or downregulating some gene. And we stratify that by distance. And you see by all the distance, we're doing quite well on predicting the sign of the eQTL. And one thing to know that even though we're doing quite well on the distal elements, distal QTL, and then the we often predict zero effect for the eClear that is quite far away, and this is still an issue. And yeah. So another thing is GWAS. We know that GWAS is genome-wide association study. It's a way to associate variant to phenotype, right? You affect, for instance, including some disease phenotype that and the variants that are associated with GWAS traits, in many cases they have some function. Therefore, they associated with some phenotype change, right? And in this case, we want to predict the sign of these GWAS elements, right? And we see that the model is on each of the target matrix precision level, we are systematically doing better than previous Bose model. Yeah, if you look at the different GWAS group, and the model is actually doing quite well on predicting the sign. And another way to predict the sign of GWAS variant is co-localize them with eQTLs, because we know the direction of eQTLs, and we see that AlphaGenome is actually complementary to resolve the sign of eQTL and GWAS variants. This is skip that. Yeah, another common task, if you think about, is linking enhancer to genes. This is a quite challenging task if you want to understand, gene expression regulation, because we know that gene expression is kind of regulated by promoter or enhancer regions, and enhancer can be very, very far away, and it's very challenging to link which enhancer is regulating which gene, because sometimes they can be, a few thousand kb base pair away. And this task is to say, can we use AlphaGenome to link which enhancer is regulating which gene, right? Because that has implication on many downstream applications in terms of understanding enhancer function and gene expression regulation. This is an ENCODE enhancer to gene linking benchmark, and on zero shot means that we don't, we don't train any additional model, just use AlphaGenome out of the box, and we see that this is doing quite well, and even slightly even better than previous model. And another way to do that is a supervised way, means that we take AlphaGenome predictions, we add a bunch of features, and we see if we can, do better by training an additional model on top for this task specifically. And we see this even slightly outperform previous enhancer to gene linking state-of-the-art model. So another type of variant is polyadenation variant. These are variants that affecting polyadenation of the transcript, means where the transcript terminates, right? We know that a transcript, when it's become a mature transcript, it needs to add a cap on the 5' and then doing a polyadenation tail in the 3'. And this task is to say, can we predict if some variant is affecting where the polyadenation happens, right? Because that will affect the UTR length, 3' UTR length. So for instance, in this case, there are two alternative polyadenation sites, right? One is from here, this is the reference. Another is here, which is with the mutation. And you can see that with the mutation, this is observed RNA-seq coverage. With the mutation, you see that the UTR is longer, basically is extended a bit, right? And without the UTR, this is shorter. Why this matters? Because 3' UTR often has regulatory elements for the gene expression, for instance, microRNA binding site or RBP binding site, right? Imagine that the length of the transcript has a lot of implication on the stability of that transcript. So yeah, this is one type of variant that, if you make the transcript unstable, as we've seen before, is the same as decrease the gene expression of that transcript. right? So this is to to check if the model is doing well on predicting polyadenation change, and we see that in general we're doing quite well across the distance. Across the distance. This is one specific example that we recaptured the observed RNA-seq coverage quite well, and we predict this. And you can see that for those polyadenation signal, this is the canonical polyadenation signal TTT-AAT, and we can recapitulate this motif also quite well. Here's one concrete example for how enhancer or is affecting gene expression, and this gene tau1 gene is an oncogene. Upregulating this gene often leads to very some sort of cancer, and in this case we show a few examples here. Actually, the variant has three groups. One group of variant is upstream of the gene, so this is tau1 gene. Start from here, end from here. This is a negative strand, and you see that here's some variant. Actually, most of them are insertions or deletions that are upstream of the gene, and this is enhancer, or the variant that actually create an enhancer. We can see, and downstream is a 3' neo enhancer, and we have some variant close to the promoter region, and you see that with all the mutations. Here's one example of that mutation, one insertion. So two nucleotide insertion on the 5' of the gene, we see that it actually affecting the histone modification of this region, for instance. And here are some activating histone marks that we see that in general this is upregulated. So this is a difference between the alternative and reference. We see the alternative is higher, therefore it's positive. And in general, these activating marks are being upregulated, and the repressive marks are being downregulated. What this causes in the end is upregulation of the oncogene. And as mentioned, the upregulation of this oncogene will lead to some cancer. And this is one example of many of these variants highlighted here as yellow triangles. And here we predict a bit more systematically, right? Here the yellow triangle are these oncogenes stratified by different groups, and the gray bars are random sampled control variants, right, within the region. And we see that the control variants generally has minimum effect compared with the actual oncogenic mutations, right? And this is to say that the model actually captures these cancer driver mutations, even though they are, in the regulatory region, they actually attribute to enhancers in this case. And we do in silico mutagenesis. Again, this is to interpret the model, what kind of features the model is using to make its predictions. And in this case, we look at the a few different tracks that the model predicts, DNAs and some histone modifications, and we see that this corresponding to this MYB motif that is quite known. So we showed some regulatory features and splicing feature, and one thing that we want to understand is, in GBAS trace, if the model is picking up the causal signal for this GBAS trace. And here is we take the trait gene data set. So this is a data set that collected some causal mutations for some traits. And we see that the causal group is enriched a lot with very strong predict very strong effect compared with a control group. The scores for the causal group is much higher compared with control group. We have more, the red bars is wider in the causal group compared with the control group. So here's some overall picture of, we're picking up causal variants, but then here's some concrete examples which I found quite interesting. for instance, this one, again, is kind of in the promoter region. This is upstream of that transcript. This is, again, a negative strand gene. And we see that with the mutation, this gene is very much no longer expressed with that mutation in the promoter region. And this is in the heart. And here is another. By the way, each of these mutations is associated with a known disease, right? Yeah, this is 4 of these plots are 4 different diseases and 4 different variants. And here's another variant in the actually polyadenation region in the UTR region, and you see that the model actually again predict much lower expression with that mutation. So if you look at the exome coverage in the red line, it's much lower compared with the wild type. And yeah, here's another variant in the I would say enhancer or promoter region, and you see that again this is much lower expressed. And if you look at the ataxic signal, this is profiling accessibility of the DNA, and you see here is the mutation make it no longer accessible. If you see the accessibility. Here's another example of the, this is a GATA motif. If you know the, and here is, again, this is much lower expressed with the mutation. So these are 4 different mutations. I think like in a typical case, for instance, I'm more experienced with rare disease. I don't think people really, look at this kind of variants in rare disease. These are regulatory variants, right? So this is just to show that the model actually can capture some of these disease-causing motifs, mutations in regulatory region as well. Yeah, what's the limitation? So we show some great examples of the model predicting splicing variants, DNA accessibility variants, eQTL variants, polyadenation variants, and many other type of variants, right? So what are the limitations of the model? This model focuses on regulatory variant effect prediction, and we see actually this is still a quite complex task, and this is far from being completely resolved. And the prediction accuracy compared with coding region, for instance, missense variant, is actually much worse. And the distal enhancer region is still quite challenging. And the model has fixed cell types. We mentioned that we have nearly 400 different cell types or GTEx tissues, but that's we have much more cell types than that, right? And the model is fixed with the number of cell types it can predict. And it focuses on variants. We haven't benchmarked the model on individual genome prediction. Yeah, and the model is trained for human and mouse only. We haven't done much interpretation beyond the individual examples we've shown. We haven't done this systematically. This is this is overview of the model. So how can you use the model? And really encourage you to give a try of the model API. It's actually very easy to use if you follow the notebook examples we give. And here's with very we give very concrete examples how you can use it. Even if you don't have programming experience, I also encourage you to try it, because it's quite easy if you do that. And we have people in the team that who do not have technical background, and they're able to walk through the Colab and make this kind of plot very easily, because all the examples are given to you. What you want to do is almost like give your own variant, your own intro you want to make prediction, and which oncology you want to predict, whether it's blood, is brain tissue, or some other cell types you're interested in, right? Yeah, so to summarize, we have the quite comprehensive, accurate DNA sequence model for human variant effect prediction, and we have very long sequence and high resolution, and this enabled the accuracy. And the variant score is very powerful, and it's actually very fast through the model API. And yeah, The API is publicly available for non-commercial use. It's also a powerful platform that we can extend to new data set and assays, or even some other species, for instance, model organisms in Drosophila or C. elegans. Here's a great team. Here's the picture we took when we launched the model API. Yeah, thanks to this amazing group of people. Yeah, thank you for your attention. I'm ready to take some questions.
Thank you. If the audience could give me join me in doing a digital round of applause for Jun on that showcase of AlphaGenome. Thank you very much, Jun. And I'm going to go to your audience questions, of which there's several. And if you would like to add to those questions, please go into the Q&A section on the top bar, and we'll be going through those based on how highly voted the question is. The first one that I can see that's highly voted is a question about head-to-heads with Splice AI, Jun. Belma Mutapchik says, given that Splice AI is widely used by clinical labs for splice variant interpretation, how does AlphaGenome compare in terms of accuracy, clinical utility, and ease of integration? Which I think you've got some metrics on the outputs there. I guess the questions would then be on the clinical utility and ease of integration. Following up with, would using one model over the other lead to discrepancies in variant interpretation between different labs and clinicians? And in what scenarios would you recommend using AlphaGenome instead of or alongside Splice AI in particular?
Right. It's quite a few questions, so maybe let's break it down. So on the ClinVar, this is one of the tasks that clinicians might look at, right? ClinVar is variant pathogenicity prediction. And here's AlphaGenome on the blue bar, and compare with Splice AI is this red bar. And you see on all the tasks, we are actually doing quite well compared with Splice AI. This is just to say that the model is, first of all, it does well on the variants, and second, I want to say that it is quite comprehensive, right? Splice AI will predict. So here's a splice site prediction. Splice AI may tell you here's the splice site with the mutation. The splice site is no longer there. But AlphaGenome tells you much more than that, right? First, it tells you that, well, without this splice site, what happens is exon skipping, right? Here's a new junction that previously was not there without the mutation. First, it also gives you RNA-seq coverage prediction, where it tells you, now this exon expression is no longer there, but the other exon are still there, right? This is the information Splice AI is not directly telling you. So no longer, not just the performance is better, but also, we give more this kind of interpretation, right? Where are the new junctions? Where are the cryptic examples? For instance, here, where are the cryptic exons, and where are the new junctions? And this is first question on the performance, and second, how easy is this being used, being integrated? We know that Splice AI provides pre-computed scores, and our model at the moment is only accessible through the model API externally. We were working on to make the model much more accessible for clinical use case. So bear with us, and this is on the usability. I think another question is discrepancy. Is that what I remember correctly?
Where might the outputs of Splice AI versus AlphaGenome be different? I guess are there edge cases where there's known systematic differences between the two models?
Yeah, for sure. I don't know whether there are systematic difference. So we know that because the model performance is better, we know that there are cases that we make correct predictions, Splice AI not. We don't have these concrete examples showing here, but there are cases like that. And I would say with all these tools for your usage, really do some homework. And obviously now these tools is really approved for clinical use directly. These are not designed for that purpose, so you always need to double check with other evidence, everything. Yeah.
Thank you. The next question that's got some votes on it is from Aaron Ventler, who asks for RNA-seq and ATAC-seq track prediction. A known limitation of this type of model has been poor context generalization, i.e. the model performance drops significantly when using it in unseen cell lines. And they said, I've seen you've mentioned this is a limitation in the slides. Could you expand a bit on how you attempted to solve for this and what you think it would take to get the models that generalize across the context better?
Yeah, so the cell types and tissues, indeed, the generalization can be very, very bad because many of the regulatory elements, they are very cell type and tissue specific. So at the moment, the model only predicts what cell types the model has been trained on. So this is kind of limitation. Hopefully, future models can address that by, some other method that can, generalize across condition. At the moment, the multitask models, none of them really generalize like that. This is still like unaddressed limitation.
Thank you, Jun. Stephen Laurie asks, first says, fantastic work by all the team and congratulations. If we submit a request to the API, are the requests stored? I presume it wouldn't be correct to submit any patient data, i.e. patient identifiable data, I assume you mean, Steve. Is there any privacy maintaining way to access the model?
First of all, we don't store anything like either the model like user input or output. But for privacy and using patient data, you always should be very, very careful and double check the, everything, like if this is, if you have the approval. Also check our FAQ page and how this is described. In general, be very, very careful when dealing with this kind of privacy concern data set. Obviously, it will be better if you have something like you can install in your local network and do your own predictions. I think we're going to release the model weights and everything, the code as well, very soon. Just overall, just be double check everything when you're dealing with this type of data.
Jun, are there any exemplars of where there has been a successful integration of this API into a variant interpretation pipeline, either in the clinical research setting that you know of? Have any teams on the ground done that integration work already? And is there any lessons to learn from that?
Not that we know, because I think mostly because of the limitations of the license. I think the license is still non-commercial, therefore we're limited to non-commercial teams. And for other teams, I think that people just generally use the API. I'm not sure that because we haven't released a way, I don't think they can install it in their own environment yet. We just provide an API that you can call.
You mentioned that there's a future plan to, you said, to release the code, and functionally that would, do you know what license that would be under? It would be
that's still a non-commercial license, yeah.
Non-commercial license. And with regards to, I think there was a question, it might have been deleted now, but what's the compute required if someone were to run it locally on a per inference basis? Do you have a ballpark figure on what that would require?
Yeah, so we actually optimize the model quite a bit. If you're running, it depends on what hardware you're running. We run on H100, so it score one variant within 1 second. So it's actually very, very fast. But you do need a H100 or something like that to really run the model.
And that's scoring for the one variant across all these domains that you've shown today.
Yes.
All right. Bing Wang asks, how do you know AlphaGenome is up to date with the latest scientific findings of the variant? Which I guess is a question relating to what's the future of updating the model with regards to new training data, and how often are you batching and retraining your model?
So far we don't we don't have this kind of plan really. But this is something that we just finished writing the paper and everything, but this might come later on. Yeah.
Of course. And well, I guess a question from me to wrap up then. Where do you see that you mentioned in your from your website on your bio that you're interested in solving critical problems with foundation models in genomics? Where do you see as the next hill to climb in this problem space that may or may not be related to AlphaGenome?
I think personally, I'm quite, if you think about this type of models, we train for human and mouse, and in general, we see that one type of information is, if it does well in other species, or if you can leverage data from other species to help you with human genome prediction, because essentially human genome, this kind of model is limited by the length of human genome. We only have 30 base pair genome length, and I mean the genome is not going to change in the near time. So if we need more data, we might need to go to other species. So that's where I think basically we need to scale the model, and looking at other species probably is one way to scale it.
Would the advent of long read in the mainstream change that?
Long read data would solve some of the challenges. For instance, for splicing, it definitely will help you. It can give you full transcript picture, and this definitely will help with splicing. It helps with one of the modality, and I think if you want to scale the model up, and you might need to collect some more data set, like other species or experimental data set, right? That can do some perturbation or things like that. Yeah.
All right. Thank you so much, Jun, for a generous use of your time and educating us about AlphaGenome. I'd like the audience to join me one more time in getting a digital round of applause for that excellent presentation.
Thank you.
We are very pleased to have next month a further edition of the Turing Genomic AI Interest Group. So please follow our LinkedIn for more information. We have two more in-person events. One more in-person event coming up this November as well, which is currently on waitlist, but you can join the waitlist for our Genomic AI implementation workshop, which will happen at Guy's Hospital in London, November the 13th. Again, look to our LinkedIn. And the other event that I'd like to signpost to you is that we have a showcase of the work that the Genomic AI Network funds and oversees itself, and that's on October the 13th, if you look on our LinkedIn posts as well. Thanks for coming to this edition, and have a